Hello, my name is Mahush Majid, and I'm from Framingham State University. My research for the past year has been on the effects of calcium concentration on the R77S gamma crystallin, a protein which is involved in congenital cataracts. Crystallins are the most abundant protein within the lens of the eye. Their function is mostly structural, but they also play a significant role in the transparency and the high refractive index of the lens. The lens stays transparent despite the high concentration of protein, and that is due to the short-range order of crystallins, which means that if the proteins were to fall out of place or change shape, there would result in light scattering, and it would affect the transparency of the lens. There are three types of crystallins: alpha crystallins, beta crystallins, and gamma crystallins. Alpha crystallins are molecular chaperones, and their role is to prevent any aberrant protein interactions. This prevents the formation of cataracts until a much older age, as it allows the lens to tolerate more age-related deterioration. Beta crystallins help with lens development and are expressed in the tissue outside the lens, and gamma crystallins consist of beta sheets and provide thermodynamic stability. Both beta and gamma crystallins are structurally very similar. They both consist of domains made up of two anti-parallel beta sheets, and both have very high thermodynamic stabilities, which may be attributed to the Greek key motif, a very common structural motif in proteins characterized by the arrangement of the beta strands into an anti-parallel beta sheet. So what are cataracts? Cataracts is a medical condition that results in the lens of the eye becoming opaque, leading to blurred vision or blindness. Mutations can affect the crystallins within the lens of the eye, which leads to cataracts, either congenital or a late onset of cataracts. So this happens when crystallins within the lens either aggregate, precipitate, or become insoluble, which causes the lens to be cloudy, and that in turn results in a cataract. Childhood cataracts are the most common form of treatable blindness in children and accounts for 1 million blind children in Asia and 10% of all childhood blindness worldwide. The R77S mutation is the mutation that I have been studying, and it is a specific mutation which results in congenital cataracts, and it is found to be the cause of childhood cataracts in many patients in India. It is a genetic autosomal dominant mutation which results in a substitution of arginine to serine at the 77th position in the gamma D crystalline protein. Arginine is a positively charged amino acid, while serine is an uncharged amino acid, and the mutation is believed to reintroduce a calcium binding site that had been previously removed throughout evolution. It will result in an increased affinity of the protein to bind to already present calcium ions within the eye. Evidence to suggest the site for calcium binding is at the Greek key crystalline fold. This is a tertiary structure for the gamma D crystalline. The highlighted region is the location of the arginine. So at that location, arginine is replaced by serine within the R77S mutation. It is believed that the calcium binding site has been reintroduced with the mutation, which leads to a destabilization of the crystalline protein. Observations suggest that the calcium binding site was lost through evolution to increase the stability of the protein, and this is seen as ancestral microbes contain these binding sites, but their crystallins are much less stable. Researchers have extracted the crystalline from bovine lenses and reintroduced the site, and they saw that there was a decrease in thermal stability. Previous research has been conducted on the R77S mutation using CD fluorescence, NMR spectroscopy, and other various techniques. And they were looking at the difference in stability and structure of the R77S protein compared to the wild type. The only difference they found was between the isoelectric point. The R77S mutation had a lowered isoelectric point. And the conclusion made by these researchers was that the mutation does not cause any structural stability changes, but the changes in the isoelectric point and surface charges could potentially affect the interactions between the gamma crystallins and other proteins within the lens. And this result seems reasonable because the arginine was a positively charged amino acid which has been substituted with an uncharged amino acid, the serine, thereby reducing the overall positive charge of the crystalline protein. And these researchers did not take into account the potential reintroduction of the calcium binding site and only looked at the stability and the structure of the mutated protein. A second group of researchers also tested the mutated protein using different techniques such as spectroscopy, calorimetry, and other stability assays to see how the mutated protein reacts to the presence of calcium. And they found that there is a decrease in thermal stability within the N-terminus domain of the R77S mutation, and a lower overall stability within the N-terminus of the R77S, which was not seen within the wild-type protein. So the purpose of my project is to test the solubility of the R77S protein at different calcium concentrations and to see how different levels of calcium affect the protein solubility as well as its stability. And in this experiment, we will be using the human gamma D crystalline protein. And the purpose is to investigate how the R77S gamma crystalline leads to congenital cataracts and whether the calcium binding site has a role in cataract formation. So this project is still currently in progress, but so far we were able to obtain a plasmid DNA containing the mutation, and this was done by using site-directed mutagenesis, and we were able to substitute an arginine to the serine, which mimics the R77S mutation. And the plasmid was then used to perform a bacterial transformation using E. coli cells. The bacteria from the transformation was grown out, and the protein was induced using IPTG, which is a chemical inducer. So this chemical inducer allosterically binds to the LAC repressor, resulting in the LAC repressor dissociating from the LAC operon, which allows for the transcription of the gene. And to see if the protein was properly induced and expressed, SDS page was run. And then the protein was purified using affinity chromatography using a nickel column, and this is possible because the plasmid contains 6 histidines. The nitrogen on the histidines within the protein are attracted to the nickel on the column through ionic interactions, which allows them to stay on the column. Imidazole was then used to elute the proteins off the column, and a desalting column was used to remove the imidazole from the protein. So to see if we were successfully able to extract the protein, we ran an SDS page. Although the gel was not the best gel, we were still able to determine that protein was successfully extracted. Each lane contains different parts of the entire process. So for example, the first lane is obviously the protein marker. The second lane is after induction. Lane 3 was after we broke open the cells, followed by the lysate, which was run on the column, and then we washed that with imidazole for lane 5, and then lane 6 to 10 were different elutions at different concentrations of imidazole in order to get the protein by itself. We also ran a fluorescence of the extracted protein. So the excitation wavelength was 295 nanometers, which was used to excite the tryptophans, and gamma crystalline contains 4 tryptophans. And the emission wavelength was between 310 to 400 nanometers, and it shows that we were successfully able to purify the protein, and this is the protein's native form. Although we were able to obtain the protein, we weren't able to run any tests on them so far. But the next step is to run a thermal stability assay, and this is to measure the stability of the protein. This will be done by measuring the fluorescence while increasing temperature using an orange dye which binds to the hydrophobic regions of the protein. So as the temperature increases, the protein unfolds, causing more hydrophobic regions to be exposed, allowing more dye to bind to them. And this causes a change in the fluorescence emission. So we predict as calcium concentration increases, the protein will be less stable and the fluorescence curve would shift to lower temperatures. UV experiments will also be run to see if UV exposure along with calcium concentration will increase aggregation. And we will also be testing for solubility by increasing the calcium concentration and to see if calcium has any effect on the solubility of the protein. And that's it. Thank you.